My name is Dr.S.Radha, Assistant Professor of Department of Microbiology, the Standard Fireworks Rajatnam College of Sivagasi. So, I am going to explain today about Agarose gel electrophoresis. So, this technique of Agarose gel electrophoresis, what I am going to discuss, it includes the principle of the technique and the parts of the electrophoresis apparatus, how it works, the mechanism, and finally, I will conclude with the applications. To start with Agarose gel electrophoresis, what is the purpose of this means? It is used to separate the mixed population of macromolecule on agarose matrix. So, it has a wide application in various fields that includes biochemistry, molecular biology, genetics and clinical chemistry. So, the parts of the apparatus, it includes started with the buffer tank. So, the tank will be filled with a buffer that is used for running the gel. The electric supply or power supply is given through the electrode, that includes the positive and negative electrode, and the gel casting tray where the agarose matrix will be filled, and this will be holded with the casting dam, and above that we will be keeping the tank lid that is covered, that is connected to the power pack. The apparatus as such, it is has to assemble, and here the biomolecule will be running based upon the charges towards the positive charge. So, the basic principle of Agarose gel electrophoresis includes the separation of DNA molecule. So, the DNA molecule is a negative charge molecule due to the presence of phosphodiester linkages, and it will move towards the positive charge anodal zone, and the migration will be based upon the size of the DNA molecule. The smaller molecule will move very fast and away from the well, whereas the larger molecule will be near the well. And for visualizing the DNA molecule, we have to stain with ethylene bromide dye, and this ethylene bromide, that is an intercalating agent that will used for staining the DNA gel. How it works, the procedure what we are going to see, it depends upon the concentration of agarose. Normally the concentration of agarose what we are using is 0.2 to 3%. For an average plasmid DNA isolation we use 1% agarose, whereas for promoter analysis and all we will use 2% or even 3% we will use. So, the size of the agarose decides the mobility. If you are reducing the percentage of agarose, then the mobility will be faster. Next, we are going to see about the applications of this technique. So, this technique forms the base of all the lab where we are using molecular genetics or rDNA technology. So, first of all, it is used for estimation of size of the DNA molecule or PCR product. So, for this we have to run a procedure of isolation, and we have to observe the result or visualize the result using UV transilluminator. So, after staining you can observe and you can compare the product size with the molecular marker. The first lane has a molecular marker and corresponding unknown sized fragment you can observe. So, this technique forms a base for genetics, that is diagnostic application, where you diagnose the disease by getting the PCR product and also the genetic fingerprinting analysis. Other application includes the southern blotting analysis, where using a DNA assay template we are used to locate the gene in the particular DNA, and northern blotting where you for expression studies we will use RNA assay template, and the expression of a gene in a foreign source we are analyzing. And when you are cloning the gene into another host, we are doing a restriction analysis, and based upon the size of the fragment that will be observed under Agarose gel electrophoresis. So, other application includes the purification of the DNA product, that is a prerequisite for any cloning experiment. Under this, from the agarose itself we can directly cut the DNA fragment and we can melt that, and we can use for purification and cloning process. So, this Agarose gel electrophoresis is the basic technique, and it is a prerequisite for all the molecular genetics and biotechnological experiments, and I am concluding with that, that is it is considered to be a very needed and accurate technique. Thank you for watching this video.